Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the previous lecture, we learned about different types of applications of biotechnology, in which we came to know about that how the biotechnology and its products will help in the human welfare, and they will upgrade the organisms, plants, and animals to deliver the best results they can. We have developed different varieties by improvising transgenic plants, transgenic animals. Now we are going to start with the molecular farming. What is molecular farming? Molecular farming is basically a technique in which the proteins which are encoded by transgenes in animals or crops, they are produced. Okay, so molecular farming is the production of proteins encoded by transgenes in animals or crops. The protein recovered by milk, urine, blood, seeds is called molecular farming. So it is a technique to produce recombinant protein, and with the help of bioreactors or fermenters. It helps to increase the milk production in cattle. The milk production of cattle, the quality of milk is improvised, is improved with the help of molecular farming. It helps to increase the growth rate of useful animals. It helps to increase the lifespan of useful animals. It helps in the large scale production of valuable proteins in milk like alpha lactalbumin, which is human protein, in urine and in blood of beneficial animals. It helps in the large scale production of valuable proteins in milk like alpha lactalbumin, in urine and blood of beneficial animals. It helps to improve the wool production from transgenic sheep. So all these are the parts of the molecular farming. If we can excavate, if we can explore, extract the desirable gene with the help of bioreactor can be modified to different compounds like amino acids, proteins, vitamins, drugs, enzymes, different pharmaceuticals. So these are the points which you have to remember in molecular farming. Now coming to next, the different other examples which we have developed from biotechnology, Brassica napus, it helps to produce herodin. Herodin is artificially prepared and transferred to Brassica napus, where it accumulates in seeds and it can be delivered with the help of the dispersal of seeds. The herodin protein is a anticoagulant which prevents the blood clotting. It is synthesized chemically and transferred to Brassica napus. Okay. Diagnostic and therapeutic proteins like production of monoclonal antibodies, peptide hormones, cytokinins, blood plasma proteins from transgenic plants and their parts like tobacco from leaves, potato from tuber, sugarcane from stem. Okay. So we take stem of the sugarcane, tuber of potato, and leaves of tobacco to deliver all these proteins. Okay, these proteins are transgenic proteins or recombinant proteins. They help in the diagnosis and therapeutics of different procedures, like diagnosis of different critical illness disorders. So monoclonal antibodies, they help to diagnose, helps in diagnosis of critical illness like cancers, HIV/AIDS, etc. Now, what are the different advantages of transgenic plants or genetically modified plants? Genetically modified crops. We have delivered pest resistant crops. The growing of genetically modified crops So these genetically modified crops, they help to produce the chemicals which act as biopesticides like Bt cotton. Tolerance. It helps to make plants, the genetically modified crops are more tolerant to abiotic stress like cold, drought, sunlight, etc. Salt. It helps to reduce the post-harvest losses, like in case of flower savar, which helps to reduce the production of pectinase, which will make the tomato soft. Mostly the crops like tomato, they get destroyed, they get rotten up because of large amount of production of the pectinases. The reduction in the post-harvest losses like flower savar, tomato variety, transgenic tomato. Prevention of early exhaustion of fertility of soil. It helps to increase the efficiency of minerals, increase the efficiency of mineral usage, increase nutritional value of food. Genetically modified plants enhance nutritional value of food like golden rice, which is rich in beta carotene, which is a provitamin. In the production of bioherbicides, like with the help of transgenic soybean and maize, roundraps, bioherbicides, alternative sources to industries. Genetically modified plants have been used to create alternate resources to industries in the form of starch, alternative form of raw materials like starch, edible antibodies, wait, edible antibodies, etc. Okay, it helps to produce the disease resistance or increase the disease resistance power. Okay, like many viruses, bacteria, and fungi, they cause plant diseases. Increase the disease resistance. And it helps in phytoremediation, in which there are many popular trees which are genetically engineered to clean up the heavy metal intoxication. Heavy metal intoxication. So these are the advantages of transgenic plants, pest-resistant crops, to make them more tolerant against abiotic stress like cold, drought, salinity, reduction in the post-harvest losses like flower, production of transgenic tomato, flower savar, prevention of early exhaustion of fertility of soil by making the plants so that they can increase their efficiency to use minerals. They help to increase the nutritional value of food, like in case of golden rice, which is made rich in beta carotene, which is a provitamin A. Bioherbicides to produce transgenic crops like soya bean and maize and roundraps, which are against, work against the different weeds. Alternative sources of industries to produce raw materials like starch, raw materials like starch, edible antibodies, okay, edible interferons, disease resistance power, disease disease resistance disease resistance power to increase the disease resistance power of the plants. So transgenic animals and plants, they are of many uses. Now coming to next slide, disadvantages of transgenic crops. As we have learned about the advantages of transgenic crops, there are many disadvantages of transgenic plants as well, genetically modified plants. Like they harm or they produce unintended harm to the other organisms. Means in the laboratory study, during laboratory studies and research, study and research, there are many plants and animals which are used. Okay, so a laboratory study was published in a book Nature showing that pollen from bt corn caused high mortality rates in monarch butterfly caterpillars. Bt cotton enriched pollen grains causes high mortality in monarch caterpillars. Okay. The monarch caterpillars consume milkweed plants, not corn, but the fear is that if pollen grains from Bt corn is blown by the or conveyed by the wind pollination to the milkweed plants, then they will be consumed unintentionally by the monarch caterpillars. So we have to be very assured about that the genetically modified crops we are going to prepare or we have prepared, it should not destroy the other varieties unintentionally. It reduces the effectiveness of pesticides. It reduces the effectiveness of pesticides. Like the mosquitoes, they develop resistance. Mosquitoes, they developed resistance against DDT, against DDT. Many people are concerned that the insects will become resistant to BT as well, okay, because of the process of the natural selection. Gene transfer to non-target species. There is a concern that the crop plants engineered for herbicide tolerance and weedicide tolerance will crossbreed, resulting in the transfer of the herbicide resistant genes from the crops into the weeds. These superweeds, production of superweeds, would then be herbicide tolerant as well, will become herbicide resistant. In animals, the disadvantages of transgenic plants are that they can produce allergies. Okay, the enzyme produced by antibiotic resistant gene can cause allergy. Antibiotic resistance genes can produce allergies. Okay. Now, the effect of bacteria of alimentary canal, the symbiotic or the natural fauna of alimentary canal means the bacteria. They can be suppressed. Okay. Economic concerns. The genetically modified crops to make genetically modified crops and to bring them to market is very costly. It is a very costly procedure. Okay. So that is why we need to prepare a genetically modified crop which should be cheap, easily available, and it should be therapeutic in action. I mean, it should not produce side effects. Okay. The transgenic microorganisms. The different transgenic microorganisms which are produced to perform or to be used in different industries like crop production and protection. Okay. There are various microorganisms which are used to improve crop production and protection, like insects by the production of endotoxins, fungal diseases by the production of chitinases, which suppress fungal flora in the soil, by production of antibiotic, which will degrade the toxin produced by pathogen, by introduction of nitrogen fixing genes in rhizobia for better nitrogen fixation, for increased nitrogen fixation. So the crop production and protection can be improved by all these methods. There are some common methods which are given, like insects by the production of endotoxins. So microorganisms can be uh, made to produce some endotoxins which can work against some insects as insecticides, fungal diseases, okay, to prevent fungal diseases by the production of chitinases, by the production of antibiotic which will degrade the toxin produced by pathogen, and by the production of the better nitrogen fixation strength by incubating NIF gene, that is nitrogen fixing gene in rhizobia. Biodegradation of xenobiotics. Xenobiotics is non-biological waste and toxic waste. Degradation of non-biological waste with the help of degraders like Pseudomonas. Pseudomonas, they are usually poor degraders, but in the presence of multiple genes, when these multiple genes are crossed or with the help of rDNA, we can help them out. Okay. So now, there are some important facts which need to be registered here, and you should note them. The first transgenic crop plant was tobacco plant. Was tobacco plant. The first transgenic cereal plants were maize and wheat. First transgenic maize and wheat. And the first transgenic plant which was marketed was flavarcibar, transgenic tomato. Okay. So now there are some common products which we have developed from the process of transgenes. Okay, so the transgenic plants like Bt cotton, like wheat, which is resistant against herbicide, Brassica nepus, tobacco. So tobacco helps to deliver a cow pea trypsin inhibitor. From tobacco, we have extracted CPTI, that is cow pea trypsin inhibitor. It show resistant against pest. It is a kind of biopesticide. So the cow pea is a strain, or it is a category or a type of pea. So the cow pea trypsin inhibitor, it helps as a biopesticide, okay, which is used by, or which is done by the tobacco as a mediator, as a transgenic plant. Okay. Then golden rice, reversible tomato, potato, bananas, many more. Production of chemicals and biofuel. Production of genetically engineered strains of Bacillus amyloliqueficans and Lactobacillus casei for the large-scale production of antibiotics. Lactobacillus casei and amylophilicans, bacillus amylophilicans, they are genetically engineered for the production of antibiotics. E. coli and Klebsiella penticola carrying genes from Z. mobilus could utilize glucose and xylose to give maximum yield of ethanol. So E. coli and Klebsiella penticola carrying genes from Z. mobilus could utilize glucose and xylose to give maximum yield of ethanol. Living factory and ethanol is a biofuel, emerging biofuel. Living factory for the production of proteins like human growth hormone, human insulin, or human insulin like growth factor, IGF, and bovine growth hormone. Okay, so these are the production of chemicals and fuels from transgenic plants. Next is transgenic animals. Production of transgenic animals with the help of carrying foreign genes. The animals which carry foreign genes are called transgenic animals, and these transgenic animals helps in the production of transgenic species, transgenic animals, which helps in the location, identification, and separation of desired gene. Location, identification, separation of desired gene. They help in the selection of proper vector, generally virus or direct transmission of DNA. They help in combining of the desired gene with the vector. They help in the introduction of transferred vector in cells, tissues, embryo, or mature individual. They help in the demonstration of integration and expression of foreign gene in transgenic tissue or animal. So these are the transgenic animals in which the foreign genes are carried to perform all these functions. Advantages of transgenic animals for the production of biological products like the human protein rich lactalbumin is produced by a milk first transgenic cow, Rosie. Biological products like introduction of DNA which codes for a particular product such as human protein, alpha-1 antitrypsin which work against the disease like emphysema, tissue plasminogen activator from goat, blood clotting factors 8 and 9 from sheep, and lactoferrin from cow. So these are the transgenic animals we have prepared, like sheep to deliver blood clotting factors like 8 and 9, cow to deliver lactoferrin, tissue plasminogen activator from goat, and alpha-1 antitrypsin to deliver the resistance against emphysema. The first transgenic cow, Rosie, produced human protein-rich milk, alpha-lactalbumin, which is enriched or which is having a maximum percentage per liter of 2.4 gram. It helps to provide the vaccine safety, like transgenic mice. They help to understand or study the vaccine safety. It helps in the chemical safety testing, like toxin or toxoid safety. Normal physiological development. The transgenic animals, they help to understand the normal physiological development, like for study of complex factors involved in growth, study of insulin like growth factors. They help to study different diseases, okay, like Alzheimer's disease, chronic diseases like Alzheimer's, hemophilia, etc. They help in the growth of spare parts, like spare parts production of heart and pancreas. Okay, replacement of defective parts. The transgenic animals, they help to replace the defective parts with freshly grown part from own cells. Production of clones. They help to produce clones. Okay. So this is all about for today. We will be meeting in the next turn to study different types of transgenic animals we have prepared. Till then, bye bye. Take care. Stay safe, everyone. Happy Deepavali.